Hi, everyone. Can you guys hear me? Maybe if you settle, it'll pick up my voice. Hi, everyone. I'm Chris Rakenna. I'm the lead organizer for Boston New Technology. Who's ready to learn about next generation AI? Definitely, I am. So anyway, we're geared towards supporting local innovation and educating the community on tech topics and helping startups launch and grow. And we have a monthly showcase on different themes to help local startups get publicity and present in front of the community. So we hope you can join. We actually have our next health tech startup showcase on June 10th, and it's posted on bostonnewtechnology.com. And you can find out about all kinds of great community events on our website as well. And with that, I'd like to just turn it over to Fidelity and the Fidelity Center for Applied Technology to welcome you for this partner event.
Hello. I'm Christine Venezia, and I work here as a program manager at Fidelity Center for Applied Technology. And I just wanted to welcome everybody. Here at Fidelity, we have an AI Club, and there's many people here from the club that are participating. And I want to welcome all of the people that are from outside Fidelity and welcome you to our what we're calling a super meetup. So we're trying to build community inside and outside Fidelity for the AI space, and so this was one of the things that we had in mind. So we're thankful for Chris to partner with us to welcome all of you here today too. The Wi-Fi, if you need it, is called Greenline. There's no password. You just log right onto it. And I'm hoping everyone will join us afterwards. This is going to go for about an hour, and then we're going to go into the cafe out here and have a couple of drinks and snacks and plenty of networking, and hopefully you can make new connections, learn something new. And with that, I'm going to introduce then our speakers, which is Sharad Shandilya, who works here at Fidelity, and Dr. Mahajan, who is joining him to go over what our topic is today. So welcome and thank you so much. Oh, one other thing, restrooms, if you need them, there's a ladies' room right outside the door here, and then the men's room, you have to go through the elevator bank and down the right-hand hallway if you need the men's room. So thank you.
Hi, my name is Sharad Shandilya. I'm part of Fidelity. Fidelity AI Center of Excellence is an organization within Fidelity. It's an initiative across business units to drive development and adoption of AI across our diverse company. And today's topic, we're focusing essentially on what the latest developments have been within the AI and deep learning space. And some of the current gaps where we can hope to see developments in the near future. And our guest speaker, Dr. Mahajan, is a neuroscientist where we can hopefully, and I think that there's a lot of overlap, we can draw a lot of analogs to understand where those innovations might be inspired from, which is, in studying nature, hopefully. So Dr. Mahajan, if you could please come up and lead the first section of our talk.
Okay. Everyone can hear me okay? So my name is Rahul. I'm actually a clinician, a neurologist at Mass General, and I also split my time at Third Rock Ventures, where I'm doing a fellowship working on neuroscience-related projects, thinking through clinical feasibility of in-house projects there. The thought of the topic today was, Sharad asked me to participate in this talk to kind of maybe talk about how neuroscience can inspire new methods in AI. And so I'll start with basically some disclaimers and caveats. So this is not a neuroscience talk. If I gave this in front of neuroscientists, I'd get laughed out of the room. The idea being that I want to give you the flavor, the kind of large themes, important points where parallels might exist between neuroscience and AIs, which can be useful for drawing inspiration. Already that's happened with some of the new methods within AI. and likely that trend will continue. And so maybe just making some broad themes, which hopefully is of some interest, and then Sharad can follow up with more of the harder AI stuff that I'm sure you're more interested in. So let's get into it. So overall, as mentioned, we'll go through lots of specific things. We'll talk about kind of some diseases. We'll talk about some pathways. But rather than getting lost in the weeds, the idea is going to be to just step back and see that there's lots of examples of things working at different scale. And we'll see that there's hierarchies of how information is processed. There's hierarchies in time, hierarchies in space or size, hierarchies in complexity. And I think these are maybe inspiration for future developments or things that Sharad will speak to. And that also that different systems, we think of sensory systems or motor systems and reward systems, really talk to each other very closely, and there's overlaps there. And so let's get into it with an example. So, it's getting harder and harder to find examples where humans are still doing better than AI. But as that bar keeps moving up, one of the current concepts is this thought of humans are able to generalize very quickly, right? We can look at a single data point. So I can look at that picture of a Segway there in the first panel. So one point is immediately recognizing it, that's a Segway, that's great. But then just based on that one data point, those set of pixels there, a human can figure out that the correct classification of that is not any of those other vehicles with wheels, but really it's that other Segway on the right side there. And similarly, using just that one data point, that picture of a Segway, the human is able to parse it into different parts, generate new examples, all from a single data point. And obviously this is still something that our machines are hoping to get to. But when we do this, really the human has an unfair advantage, right? So we're not doing this based on simply the pixels in front of us, simply the image that's coming in. So yes, I see the pixels, and maybe I can, in a simple association, come up with the word Segway based on the pixels there. But really, for me to do all of those calculations and generalizations that mentioned on the last slide, there's a lot of previous knowledge and learning that's being deployed in doing that. And so that previous knowledge and learning is one of those examples of a hierarchy, right? Of learning that's taken place at different scales, at different levels of time, and then we'll see in a little bit that also in different levels of space within the brain itself. And so when I see that image, there's activation of concepts and memories learned. So, Segway reminds me of motorcycles, of the idea of transportation, of things with wheels, of controls and joysticks. Those are concepts that I had previously learned based on my experience and decades on Earth, right? And for me to really understand those concepts, before that came concepts of, learning to walk, learning how physics on Earth works, learning that there's such a thing as balance. I have a mature visual system that let me kind of capture all this information. And all of those systems really, if we step farther back, evolved over the course of generations, over the course of thousands of years, millions of years, on evolutionary time. And so really we're impressed with the performance at the last second, but the learning that allowed that to happen took place over the course of days, decades, and generations before. So we'll, having seen that, we'll kind of go through each of those steps. So we talked about learning occurring over the course of generations, over the course of evolution. So I'm going to sum up nervous system evolution in one slide here. And really, the human brain in its current form evolved from simpler structures in the past. That's actually not a technically correct thing to say, but like I said, we're going to suspend that so that we can actually get to some themes. Brains in general started with just nets, right? So undirected kind of nets of neurons, seen in the top right there in a creature called a hydra. And ultimately what our brain is doing is it's optimizing our behavior for the environment, right? So it's optimizing a reproductive success. And this reproductive success is a function of our motor output. So all behavior at the end of the day, comes down to motor output. It comes down to how you're moving or how you're interacting with the environment. And the ability of the brain to do this has increased in complexity over time. And really basically showing another evidence of hierarchy here. So we start with sponges that actually don't have any neurons. They just have the motor output. So they just have essentially muscle cells without any neurons controlling them. You get to sea anemones where you have muscle cells and motor neurons, the neurons that control the muscles. You get to hydra, where now you have sensory neurons. You're now getting input from the environment, which informs how you are able to control your behavior. And then you get to increasingly complex brains. In vertebrates, now you introduce interneurons, so there's actually a processing layer between your sensory input layer and your motor output layer. And finally, you get to vertebrates, where you have large brains, really neurons that are clumping together into areas we call brains, and you can kind of see that in just the shape of these structures. The takeaway from here is that nothing really radically new happened between the sponge and us, except for a change in architecture, right? We didn't develop a new type of machinery, a new type of neuron. We developed, it's really changes in scale, so there's more neurons, and there's changes in their connectivity to each other and the architecture of that network. But fundamentally, if you go to a squid or a sponge, their neurons are made of the same ion channels, the same cell membranes, and a lot of the same fundamental machinery that ours are. All right, so that was the evolutionary part. Now we step up to the next part, the next kind of scale, and that's the human development, so within our lifetime, right? So in utero, you can see some figures there. Basically, there's a hardwired programming in our DNA that leads to the brain as it looks like in a newborn child. And when you look at a newborn child's brain, it fundamentally, on a gross level, looks like an adult brain. All the lobes are there, your frontal lobes are there, your temporal lobes are there, your cerebellum is there, the brainstem is there. So all the big parts are there. And so we all start out with that same kind of global structure. And so that learning, the fact that that global structure needs to exist, that information exists in our DNA. So really the DNA contains a form of learning which took place over the course of generations. And really, we have that head start. Our networks have been primed over the course of millennia, from which we start our learning within our lifetime. So there's scales going on. So at birth, the brain looks like an adult brain, but obviously it isn't the same as an adult brain. They're at the kind of lower levels, at the microscopic level, all the systems are immature. And the idea is that within the first few years of life throughout your childhood, the brain is developing rapidly. And the way, again, this learning and development happens is not through sprouting new neurons. That happens maybe for the first year or so. It's through changes in connectivity. And so the changes in connectivity occur at different scales. So there's something called myelination, basically making connections faster between certain neurons. And while I talked about certain development being hardwired in the DNA in terms of the global structure of the brain, there's other development that occurs that's not hardwired, right? So we know that if a child misses a critical period, certain types of development will not happen, right? So if you were blindfolded and you did not have visual stimuli growing up, and then you were unblindfolded after a certain age, you would not be able to see better no matter how much training data you got, no matter how much experience you got in the world. Meaning that for that development to occur properly, it actually requires input from the environment. It's not hardwired in the DNA. So some types of learning is hardwired in the DNA, and then other types of development and learning actually occur during the lifetime and and require input from the environment. So we got through development. Let's talk about learning. Now we're getting to shorter and shorter timescales. So learning. In the brain, there's when you're learning a new task, when you're doing something, interacting with your environment, learning always involves changes in the brain. And so every time you learn something, the structure of your brain is changing. It's just a matter of what scale that structure is changing. So you can change the structure in terms of the shape of a protein and the activity of a protein on the order of seconds, probably happening millisecond, right? Some ion channels activity is increasing, decreasing within a synapse. Other types of structural changes, right, the formation of a new synapse, so one neuron reaching out to another, forming a new connection, that takes, it can actually happen over the course of a few minutes, but it really takes several hours for that new connection, new synapse to mature. And then things like myelination, which I talked about, this speeding up of connections between brain areas, that can take weeks to months to occur. And again, really, it's not about formation of new neurons. It's about changes in connectivity and how easily signals are passed from neurons one to another. So we've talked about evolution. We've talked about development through childhood, so the scale of years. We've talked about learning. And now we can talk, kind of finishing out that example of the segue, right? We're working through the different columns. We can get to the part about just recognizing images, right? So the visual system is kind of a famous example of, again, a system where inputs are processed in a hierarchical way. So it's been shown in monkeys, and there's parallel information in humans that as you go through different layers of visual processing, shown in that diagram there, V1 in the back of your head here is where the neurons from your retina, so your photodiodes, really land here in the back of your skull. So that's in the back of your brain. So that's the very first input layer where those photodiode neurons land. And with subsequent layers going down to V2, V4, you're actually computing more and more complex features. You detect edges in V1, shapes in V2, objects in V4. By the time you get to the inferior temporal lobe, you're detecting faces, objects. And we have evidence of this in humans. It's kind of interesting from things like seizures. So people who get seizures in the back of their brain, basically those neurons are firing abnormally, and they'll see colors and lights and patterns. And depending on where the seizure is coming from, the complexity of those hallucinations changes. So if it's happening in V1, you see very kind of elemental squares, triangles, dots. And if it's happening way out in inferior temporal lobe, you can hallucinate faces, animals, and some of the things described there. I don't know how many people in the audience have migraine. Very common to have migraines. A migraine aura is essentially a similar thing, where you have abnormal activation of these neurons giving you these visual hallucinations. So we talked about hierarchy, kind of with the example of that segue going through kind of lots of different scales. Another example of hierarchy is in how our motor system works. So at the simplest level, and I do this in the office as a neurologist, is you can tap on somebody's joints with a hammer and you get a very simple reflex. So that reflex actually involves one synapse. So it's the neurons that detect the stretch going to your spinal cord. They don't even go up to your brain. It makes one synapse, and the motor neuron comes back and actually causes the muscle to contract. So that stretch, the reason we do it is to just check the wiring all the way back to the spinal cord is working. So we know all of that section is working okay. So that's kind of the simplest input in terms of a stretch reflex. But the stretch reflex can be under the control of a more complicated pattern generator. So when the bicep contracts, there's actually a inhibitory kind of motor pool that makes the tricep relax, right? So you're getting slightly more complexity. And that pair of actions is under the control of a secondary pattern generator, right? So when you touch your arm to a really hot surface, you withdraw. Now that's multiple muscles working. And that can be under the control of a higher, more complicated pattern generator. And so I've just listed out some of these until really you go physically up into the brain and you get into areas that can initiate these patterns. And you can get to pattern controllers. And so the pattern controllers are up in the cortex, right, into the actual higher parts of the brain where you're doing conscious thought, volitional things. And there you can make a decision to move, and that can percolate all the way down this hierarchy, and you can get a fairly complex movement initiated at the top. Now just some kind of general comments. So in terms of memory, there's lots of different types of memory. This is a busy slide, but really the takeaway is there's different kinds of memory. And memory can also be broken down in terms of hierarchies and subdivisions. So you have extremely short-term memory, you have a medium-term memory, and you have remote memory. And the thought is that different parts of the brain are kind of doing main tasks in some of this. So any kind of memory involves encoding, so actually writing things down, storing, and retrieval. And the thought is that the frontal lobes are kind of the file clerks, keeping things organized, helping you retrieve them, and the medial temporal lobes of the hippocampus is